Well, hello everyone, and thank you for attending today's discussion. A few housekeeping items before we get started. This webinar is being recorded and will be sent to your email in the days following the event. If you have questions during the event, you could submit them through the Q&A tool below. And we obviously have a robust participation today, so we'll try to get to as many of your questions as we can during the next hour. So I'll be fielding these questions throughout the webinar. And in addition, we will also be asking everyone to answer a few live polling questions during our discussion. So please keep a lookout for those, and your participation will be greatly appreciated. So let's begin. My name is David Wicks. I'm Vice President in our corporate platform groups at NASDAQ, and I'll be your moderator today. NASDAQ's corporate platforms division is the group that works with our listed companies and IPO prospects and really empowers our companies with technology and intelligence to innovate, grow, and stay ahead of the changing markets. We offer an integrated suite of market intelligence, analytics, and collaboration solutions that support business leaders around the globe. And we do get to work with true innovators in the marketplace, companies like Certara. And joining us for today's discussion is Julie Bullock, where she is Vice President and Global Head of Clinical Pharmacology and Translational Medicine at Certara. So Julie, thanks so much for being here and lending your expertise. And before we actually jump into the questions, would you like to take a minute and just introduce yourself to the audience?
Oh, sorry. Yes, absolutely. Thank you, David. And thank you to NASDAQ for inviting me to speak today. Starting from the beginning, my educational background is in pharmacy, and I was introduced to oncology, drug development, and clinical pharmacology during my postgraduate years. I started my career at the Food and Drug Administration and had roles as both a clinical pharmacology reviewer as well as team leader and CEDR. And I was at the agency for 10 years, the majority of which was in the oncology review divisions, reviewing both small and large molecule IND, NDA, and BLA applications. After leaving the FDA, I started consulting, and I've been advising both biopharma and biotech for the last 7 years on a variety of development, regulatory, and clinical pharmacology issues, focused mostly in oncology therapeutic area, but I have experienced other therapeutic areas since leaving the agency. And to date, my career has really been dedicated to optimizing oncology therapies and development, both inside the agency and now on the outside. So it's wonderful and a very unique opportunity to be a part of this important discussion on Project Optimus today.
Thanks so much, Julie. And let's jump in. And let me just start by saying I think we can all agree that drug developments in the oncology field are crucial to the success of patient care. And just recently, the FDA's Oncology Center of Excellence launched Project Optimus, the much anticipated initiative to reform the dose finding and dose optimization paradigm across oncology by educating, innovating, and collaborating with companies, academia, professional societies, and international regulatory authorities. So their goal is to address issues relating to dose optimization in clinical trials, assessing the safety and efficacy of oncology drugs. So historically, the dosing strategy for oncology drugs has focused on the maximum tolerated dose, which I think is a good place to start for today's discussion. So Julie, can you give us a bit of background on the maximum tolerated dose paradigm? And also, what is it and what does this treatment mean for patients?
Yeah, sure, David. So the objective of the 3+3 design is depicted on this cartoon if it's showing for the audience. And it is to escalate dose upward in cohorts of 3 patients until 2 out of 3 patients experience a dose-limiting toxicity or a DLT during the DLT period, which is typically the first 30 days of treatment. These DLT definitions can vary depending upon the drug class and the patient population, but usually these are defined as fairly severe toxicities that require supportive care or withholding of the investigational treatment. And once the dose level is depicted on this slide where the DLTs have been identified, the next lower dose is determined to be the maximum tolerated dose. However, this MTD dose is not a toxicity-free dose. It's just a dose that didn't cause dose-limiting toxicities in 2/3 of the patient population. So, the MTD, a dose determined by safety alone, is often the dose that's taken forward in subsequent studies. And it's depicted here in this slide. And this is a relatively conservative slide. I've seen multiple indications, multiple combinations that just take this dose and run with it throughout development. I think that this often means that patients are treated at doses that cause toxicity but have not necessarily been determined to confer benefit than lower doses of the drug, which may not have toxicities. So that's, some of the main, the overview of the MTD approach and getting a little bit onto, some of the issues with MTD.
Yeah, that's great. And as you kind of think about the MTD paradigm for oncology drugs, what are some concerns there?
Yeah. So for a cytotoxic drug, the MTD approach makes some sense because these are not targeted drugs and they exert effects on all rapidly dividing cells. And that's the historic way that we've been developing drugs, for many, many years in oncology. But the contemporary oncology treatments, the biologics, the targeted agents that we have been moving towards those more targeted, specific approaches. They're not just targeting all rapidly dividing cells. So giving excess drug over that target makes not very much sense. And you should dose technically to our target, and then we should stop, because giving doses that are higher usually only provide for additional exposure, which will potentially result in off-target toxicities and very likely no additional benefit. The other limitation with the MTD paradigm is that DLT criteria window. So I mentioned earlier that window is usually the first 4 weeks, or 30 days or 28 days, depending upon the protocol. And it's limited to that one cycle. And so while this applies nicely to cytotoxics, which are given sporadically every week or every 3 weeks for short-term periods, it's less relevant for many of the targeted agents, which are given frequently, sometimes daily, for longer periods of time, usually until disease progression. Therefore, these targeted therapies, dosing based on MTDs from DLT during the first 28 days of therapies is not as informative as it could be. And I think this gets to the issues that the FDA has been raising for a few years about the relative tolerability, especially the long-term tolerability and tolerance of the persistent low-grade toxicities are definitely an issue for patients. And they deserve more thoughtful approaches to dosing. And I think this is where the FDA sees the need for change. I've been on the FDA side of the table and I've been on the other side of the table. And it's clear that patients that are unable to tolerate therapy past first cycles, that they still respond after receiving multiple dose reductions. But when I was at the FDA, unless those lower starting doses are evaluated in clinical development, it's exceedingly difficult to retrofit those early dose findings into an optimal dose at the time of approval. So, I'll close this by saying that I'm not suggesting that 3+3 design should be retired or that it's dead. It has its utility. It's a very fast and efficient way to escalate doses for further testing in the clinic. But we have to really, as a community, rethink how we utilize this data coming from this study and how we can explore more ways than just using 3 subjects to make decisions across the dose range to obtain information we need to better optimize dose.
Great. And Julie, that brings us to our first poll question for the audience. So we're going to post that right now. And the question is, are you using maximum tolerated dose as your guiding principle for oncology drug dose selection? So we ask that you select one, and then we'll give it a minute and see what the results are. All right, and maybe we can see what the results are from the poll.
That's about what I would expect. Yeah.
So I was going to ask you if that surprised you, but it sounds like it's kind of what you expected.
Yeah, it is. And, even for the partners that I work with and the clients that I work with that are using other approaches, they sometimes bring in these sophisticated kind of CRM methods or Boyne designs, but when you really dig into the details, they're still 100% focused on safety. So I'm actually curious as to whether or not that 28% that says we're using another approach, if they really drill down to their other approach, like how much is weighted towards safety versus other components of the development. So yeah, this is really interesting. Thanks for sharing, David.
Yeah, that's great. And thank you everyone for participating. And we'll take a question or two from the audience. And let me just pull one of these up. Just bear with me. So first question, is there a specific draft guidance available on Project Optimus? And this question says, person that asked says, I didn't find one on the FDA Project Optimus page.
Yeah, and I get into this a little bit later in the talk, but we can go ahead and revisit it now. So the Project Optimus page, which was, I believe, went live a couple of weeks ago, does not have a specific guidance. And I actually am not sure whether or not the FDA is going to be issuing specific guidances with regards to Project Optimus, because if you really think about it, dose optimization and identifying the optimal biologic dose is not a pan activity that is applicable to every single agent in the same way. Every therapeutic agent is going to have a different approach to dose optimization and what they use and what they leverage. And so the agency has put together, they cited 3 guidances. One was the population PK guidance, the other one was the exposure response guidance, and the last one was the 1996 dose response guidance. So that one's been around for a really long time, and they had to, unearth it from the archives and dust it off, but it's relevant. It's used by every other therapeutic division, and so I think the time is now that the oncology division starts utilizing this as well.
And Julie, let's take one more question before we go on. So the question is, how do we know the systemic concentration is sufficient to hit the target at an effective concentration?
Yep. So that's I get that question a lot. And the answer is that we don't. But just because we don't know whether or not the systemic concentration is hitting the tumor doesn't mean that we shouldn't do hard things. And I think that a lot of the time is, well, we don't know what the tumor concentration is, therefore we have to ratchet up the systemic concentrations to cover ourselves. And I think that, you can do that. You can definitely see whether or not these higher doses or higher exposures are conferring additional benefit. But that's basically what the FDA is asking us to do is, if you think that you need to go higher, kind of prove it, show us that these concentrations are needed. But yes, this is the #1 hurdle, especially for solid tumors. It's a lot easier in the heme malignancy world when you're dealing with, the hematological type things, which are a little bit more relevant to systemic concentrations. But yes, it is an issue, but I also, I think that it's a great opportunity for the entire community to come together to figure out how we can figure out better ways to correlate systemic concentrations to tumor concentrations in ways that we've never been able to that haven't been able to do before.
Great. And Julie, now that we have the background on MTD, let's talk specifically about Project Optimus. So let's start the basics. What is it? Who's behind it? And ultimately, what's it trying to achieve?
Yeah. So you touched a little bit on it in the introduction, but Project Optimus is like all the other oncology center of excellence initiatives, such as Orbis, Equity, and Confirm, which have been developed recently based on the contemporary needs in the oncology, both treatment as well as development space. So it affects patients as well as the regulatory and clinical development. So, the basic goal of Project Optimus is to reform dose optimization and dose selection paradigm in oncology drug development. And as you mentioned earlier, it's going to be accomplished by educating, innovating, and collaborating with a variety of stakeholders, including patients, to move forward more sensible dose finding and dose optimizations that really focus more on efficacy instead of just focusing solely on toxicity and safety. And I've been fielding, as mentioned earlier, that a lot of questions as if the FDA is going to come out with that token guidance, the one that guidance is going to give us all the answers. And I think that, while we may eventually work to that, I think it's going to take some time. And as I mentioned, they have put the 3 guidances on the webpage, which I definitely encourage everyone to go read. And then, in the meantime, I think that the 2021 Friends of Cancer Research Annual Meeting white paper was really comprehensive and gets to a lot of these issues that I'll speak of today. And the October 2021 New England Journal of Medicine by Dr. Shaw and OCE colleagues those are the 2 publications that, until we get some specific guidances, people should really be focusing in on.
Gotcha. So I think a logical question is, why now, right? Why the sudden change in expectations?
Yeah, this is an interesting one, and I'm sure, if we all could gather over, at a bar over some beers, it would be really interesting to hear why other people think why now. And I have my theories, but I'm no longer inside the agency, so I can't speak to the specifics regarding the development of this and why now. But I can say that the topic of dose and dose finding has been a recurring agent for the agency topic going back to 2013 when I gave a talk on targeted agents when we were having some issues with one of the some very concerning toxicities reported for a recently approved targeted drug. When I was getting ready for this talk, I went back to a lot of the other targeted agents and was looking at, just like, why are we approving all these drugs if the majority of them were having all these post-marketing requirements related to dose finding, and also just going through and seeing that, all these drugs had, 50 to 60 to 80% of the subjects who were not even on the starting dose by the time they either left the study or by the time of their first response. So it's like, well, if we don't need to start there, then if we're constantly reducing dose, then why are we starting so high? And I think that, since 2013, there has been numerous workshops sponsored by the FDA, ASCO, AACR, Friends of Cancer Research. So this is not a new topic. There's been multiple publications. This one right back here is one that's just filled with dosing and oncology. And I think that Project Optimus is an accumulation of these years of learning and discussion, but I think it carries with it a large bag of frustration from the FDA, as none of these workshops or publications since 2013 has led to any meaningful changes in the way companies evaluate dose and development. And so, therefore, the Oncology Center of Excellence needed to provide a framework to reset expectations for developers. As to why now, I'll talk about my remunerations on this a little bit, but I think that the field of oncology is getting relatively crowded. I don't want to say it's an easy place to develop drugs, but it is relatively easier than other therapeutic areas. And I think that by imposing companies to slow down and think about optimal dosing, there will be better checks and balances to ensure that drugs worth developing are moved forward while curbing the less promising agents. So right now, any drug that has identified a maximum tolerated dose moves forward in development, regardless of efficacy. And that's concerning. And I think that we've hit a saturation point, not only for patients, but for resources, both inside the FDA and on the outside. So I think that, maybe it was just an accumulation of the years of seeing all these things happen and nothing changing. And the agency thought that Project Optimus would be this is finally now, 8 years later, it's a good time to start talking about this and doing something about it.
Yeah. Well, that's great. And just a quick note to the audience. I know some questions are going into the chat, which we'd like to avoid. So there is actually, right next to the chat, there is a Q&A button. If you can put your questions in there, it'll help us organize and answer those. So just a reminder on that. So Julie, in my spare time, I'm a drug developer. So what does this mean for me? and what is it? What should a drug developer expect now?
Yeah, absolutely. So this is a great question, and I think the one on top of everyone's mind lately. And I think if there's one thing that people take home today from this webinar that we need to everyone needs to hear, is that this 3+3 MTD approach to dose finding is no longer going to be an accepted route, and that we really need to begin to reset our focus on providing efficacy across the dose range. And by doing this, it very well may change the landscape of the current development program. You know, as I kind of hinted at earlier, you're not going to be able to move from dose escalation into multiple expansion cohorts and multiple combination cohorts as rapidly as we have in the past. But that actually might be a good thing, because it's kind of it's turning into a slight chaos and resource burden for a lot of people. You know, getting back to the expectations, though, in this slide, I think the agency is looking for integrated analyses to support dose justification, and this entails looking at dose and exposure response relationships, and looking at all the data in addition to efficacy and safety. I have on this slide a couple of topics with relation to toxicity. You know, I mentioned earlier that you should be critical of all adverse events. You should not just focus on DLT criteria and DLT windows, and that we must start considering low-grade events that affect patient quality of life. And, you know, these types of things, the patients aren't going to take your drug, they're not going to benefit from your drug. And so, you know, we need to make sure that patients are able to tolerate your drug and stay on for long-term efficacy. You know, and this will be for the investors, and I've learned since being on the outside that it's the investors that really drive development, unfortunately. It's not the science sometimes. And so the investors need to expect that current milestone targets may not be achievable. You know, I've had conversations with clients that were like, well, we don't have time for Project Optimus because we need to start phase 2 because it's our milestone contract with our investor. I'm like, I don't care about that. I don't think the agency does either. You know, so we need to, you know, maybe go back and look at our milestones and our, you know, when payouts and when what are these benchmarks that we have with regards to our investments, because they're not going to be on the same track as they were prior to Project Optimus. You know, and if people continue to pressure science teams and development teams to continue without addressing the considerations of Project Optimus, you're basically taking that program and ratcheting up the risk of clinical holds or complete response actions, which I don't think any investor really wants to do. So this will affect a little bit of everybody, I think, and hopefully some of the investors in the audience have heard that warning.
Yeah, that's some interesting comments for sure. And as we kind of talk through this, I'm kind of thinking, you know, if this will actually hinder the pace of oncology drug development and ultimately, I guess, a patient's access to therapies. Yeah,
and you know, this is one where I think you know, there's a couple of reasons for why we probably haven't had any sort of movement towards this anytime sooner. And I think that the speed and the access has been the two main issues. And I'll talk about it later, but also statistics is probably another reason why we haven't done anything until now. You know, but I think that while this does slow down development, I think that it's still a net benefit for patients, because I'm not 100% certain that the way that we're developing drugs now is best for patients. I really do think that in some cases it's irresponsible, and that the true benefit of some of the drugs that are in the IND stage, that are in development, are seriously questionable. And so we're not benefiting patients by just shunting things forward because they have identified a maximum tolerated dose. We really need to focus on efficacy. And I think this is where the agency is really coming from. You know, the real impetus behind Project Optimus is to improve patient care and benefit at the time of approval, which means that this needs to be done in the pre-market setting, which historically dose finding has been happening in the post-market setting. And from a commercialization standpoint, that's not ideal, because if your dose changes post-marketing, it affects your pricing. And so it's actually better if you slow down and get it right the first time, instead of being forced to do it in the post-marketing setting. I think that given time and learning the approaches with Project Optimus will be benefited from. Clients will recognize that long-term, the understanding of understanding the efficacy of our different dose levels is incredibly important. And this actually is nicely highlighted in that New England Journal of Medicine, and I'll touch on it a little bit later on the benefits of bracketing. But it does improve care by decreasing toxicity. It maintains efficacy and allows patients to benefit from treatment for a longer period of time. And on this slide underneath the focusing on tolerability, I have provide access to patients with poor performance status. So by developing drugs that are front-loading toxicity over efficacy, we're taking an enormous pool of patients that are frail or unable to tolerate these new approved agents or these investigational agents, and we're not allowing them access because they won't be able to handle the toxicity. So if we think about being balancing the toxicity, if we don't need to drive to those toxicities and we can still have efficacy, then I think that we've just opened up our pool of patients a little bit more. We've allowed for broader access to clinical trials, and we've also allowed for a larger commercial market once you've been approved. So this is incredibly important. With regards to the more is not better, this is definitely going to take time, and this the people that are struggling with pivoting to Project Optimus are going to slow down the market even more. The people that have accepted the fact that more is not better are going to be able to lose less time over the course of the development period. But right now, I think that a lot of people are still very stuck in the more is better paradigm, and they do not understand that more is not better. And it will take a little while to get to that point. And I have a lot of people that argue with me about that, like, you can't take this dose if it's not efficacious. And the agency is not asking you to take lack of efficacy forward. They're asking you, to look at efficacy across a dose range to see whether or not you confer any additional benefit by giving higher doses to patients.
All right. And Julie, that leads to our second poll question. So for our audience, the question, if we can post, is what do you think will be the major impact of Project Optimus on oncology development programs? And we just asked that you pick one, and we'll give that a couple of seconds for everybody to vote. Then after our poll question, we'll take a few more questions from the audience, but give it another few seconds. And do we want to post the results?
Yeah. That's, it's the timeline, I think, that really is impacted. And then I talked about the streamlined development. So having people focus more on developing a drug for a certain indication and a certain combination instead of doing what we're currently doing, which is just tossing these very large nets out there. We're going to do, colorectal, lung, melanoma, hematological malignancies, and then we're going to combine it with, the state-of-the-art PD-1s, or we're going to develop our own PD-1 and combine it with that. I mean, sometimes I look at some of these development programs and I get slight panic attacks just trying to figure out how some of these small biotechs could even possibly do all this work. So I think that it doesn't surprise me that those two are the top reasons. Yeah. So
this may be a simplistic question, but does an increased timeline always imply an increased development cost?
I don't necessarily know if that's the case. I think that you have a tendency to, instead of spending a lot of money in a short period of time, you just spend a lot of money over a longer period of time. So I don't necessarily think that the FDA is not they are asking you obviously to evaluate more patients. And so that will increase development costs a little bit, but it's not like they're asking for additional studies. There's no additional study burden here. It's just usually increasing the amount of patients that we have in development to make dosing decisions. Yeah. But if you streamline your development program, is that cost then dumped into the other programs? I think it's going to balance out from a cost perspective. Yeah.
Yeah. So a question from the audience. Does Project Optimus encourage more use of in silico biosimulation to determine or at least narrow in on optimal dose?
Yeah, and there's a question about this a little bit later when we start talking, about what we're, path forwards and stuff. And I think that the in silico modeling it has not been, there was no guidance on that really brought up to the FDA's on the Project Optimus webpage. I do think that there are uses for in silico modeling, especially quantitative systems pharmacology. I've seen it used to really hone down on, step-up regimens or other regimens to avert early cytokine release syndromes for some immunotherapies. And I think that those types of modeling tools can be really good so that you're not spending months and months looking at different regimens in the clinic. Instead, you do that in the in silico model, you get a better idea of where you're going, and then you obviously take a thoughtful approach into the clinic instead of doing a bunch of kind of chaotic stuff in the clinic. So yes, I do think there are options for that.
So another question, at what point in development should sponsors engage with the FDA regarding dose optimization? And what form should this take? Type C meetings or others?
So the outcome of the Friends of Cancer Research meeting is that the FDA is looking as early as pre-IND to have conversations with how you plan to optimize dose. So, they are looking at this in the pre-IND, and they are looking at it in your first in human study when you submit your original IND. So I think that we have a lot of opportunities in those very early stages to pressure test whether or not the agency agrees with your approach, and those should definitely be utilized. The pre-IND is obviously a type B meeting, but could you submit a type C meeting after you get through dose escalation to talk about what dose levels you want to take forward in an optimization strategy? I think that yes, the agency would be very open to that.
All right. Well, Julie, you've definitely shared a lot of helpful information and useful context. So looking ahead, I'm wondering, do we have any information on what the FDA is expecting for dose optimization?
Yeah, so, and I have a slide on this, and this is just basically a summary from the Friends of Cancer Research annual meeting. And so without any formal guidances, all we have is to really take things out of that meeting. And I obviously have the ability to see what's happening in the IND correspondences that I see that cross my desk. So I am seeing some consistent trends from the agency with requests to establish therapeutic windows for activity and efficacy. I am also noticing that the FDA is, looking back and leveraging earlier non-clinical data, which is often not looked at once we have clinical data. We kind of we gather it to show proof of hope, but once we have proof of concept in humans, we don't go back to the non-clinical settings to see whether or not what we're doing in the humans is aligning with what we saw in the tumor growth inhibition models or other non-clinical evaluations. And they're really using this non-clinical data to narrow the range of doses for further clinical evaluation. And so my last bullet here is something that I think lacks a little bit of clarity still for myself as well as many developers is what biomarkers or PD endpoints can be best used to understand dose response. And I think that this is going to be compound specific, so I would not expect a blanket guidance from the FDA on these endpoints. The three that I have listed here, you really can't go wrong with. We're all very familiar with RECIST criteria and tumor dynamics and the clinically relevant biomarkers that have been established by like minimal residual disease. But, can we leverage pharmacodynamic or other sorts of data in our dose response and exposure response to justify the recommended phase 2 dose is still yet to be determined. And again, I'll say I think it's going to be very compound specific.
Gotcha. So how should model-informed drug development be leveraged to support your dosing strategy?
Yeah. So the three, I mentioned the three guidances, and all three actually have model-informed drug development baked into them. So two of them are solely model-informed drug development guidances, the POT-PK and the exposure response guidance, and then even the 1996 dose response guidance. It mentions using and leveraging concentration response data, which is what I have here on this cartoon here. So on the x-axis, we have dose or exposure. You could use either. And I have markations for the effective dose. 50 could also be the effective concentration. That's where you get 50% of your response. And then I've also just kind of drawn in a line for the maximum tolerated dose. And you can see here, based on this curve, and all curves are going to be different. I've seen curves that go U-shaped. There are curves that keep going up. This curve though plateaus, which means that as you increase the dose or as you increase the exposure, there's no additional benefit from an efficacy perspective, but you do confer additional safety risk. So, when I was at the FDA, this is how I looked at data. This is how they look at data. This is how we all should be looking at data. And these could be modeled data. It could be observed data, but these graphs need to be made. They need to be looked at. The FDA is expecting these types of analyses to come in with your dose justification. I did mention earlier that another model-informed approach could be use of the quantitative systems pharmacology, which uses those in vitro as well as in silico, and takes your in vivo data as well as using some of the in vitro and in silico methods to model out different dose regimens or dosing considerations. And this can be leveraged both from the non-clinical side as well as mixing non-clinical and clinical data. I don't necessarily know if those types of approaches would be used to justify the dose to take into efficacy, but it could definitely help you narrow the range of doses that you need to explore for efficacy.
Okay, and Julie, let's wrap up with one last question before going back to the audience. What would you advise developers do to get ready for this?
Absolutely, yeah. So, like I said earlier, the best advice is for everyone to just get to acceptance as quickly as possible that Project Optimus is the new reality, and to start planning to compare at least 2 dose levels for efficacy in your clinical development program. One of the really interesting take-homes from the Friends of Cancer Research, and I said, earlier in the call, like, what are some of the reasons why I think this hasn't happened until now? And one of them was statistics. And I kind of joke about it, but I actually do think it's one of the reasons why we've never really optimized dose well in oncology, is because everyone's so focused on powering this assessment. It doesn't need to be powered. You can see relative trends without a p-value attached to it. And so I think that, and that was really clear in the Friends of Cancer Research meeting, is that these cohorts, these dose optimization cohorts, need to be sufficiently sized to understand the shape of the dose exposure activity relationships, and they do not need to be powered. So as much as I love statisticians, their utility in the phase 1 world is really, can be incredibly hindering. I think that, another thing that people need to do to get ready is to really understand that dose optimization occurs in 2 stages. The first stage, you're deciding what dose levels you want to take forward to get more efficacy information from. The second stage is determining from those 2 dose levels what dose level you want to take to efficacy studies. Everyone's blurring this. They're hyper-focused on one stage versus the other stage, or they think it's a one activity exercise, and it's really not. It's 2 parts. And the rest of the slide kind of gives some tips from the Friends of Cancer Research meeting that, I think are relatively logical with regards to dose levels that we should take forward. So you should not take dose levels that have overlapping pharmacokinetic exposures. And while this seems incredibly logical and obvious, you would be surprised at the number of people that come to me that say, we're going to take these 2 dose levels forward. And I'm like, that's great. They provide the exact same exposure. So why don't we think a little bit about this? the lowest dose should be the minimal dose expected to provide activity. The FDA is not asking you to take an ineffective dose forward. And, if you are not getting the activity that you want from this low dose level, then allow for intrapatient dose escalation. Usually the highest dose is going to be chosen with safety in mind. And that will typically be based on the MTD criteria, which is fine. For the analysis, again, I can reiterate again that they need to be integrated, and that the results from this quote stage 2 dosing study that we go through will be the one that takes you to your registrational trial. And the bottom box here highlights that if the efficacy and safety are similar between the lower and the higher doses, the lower dose should be selected as the final dose for registrational trials. And this came out really clearly at the Friends of Cancer Research meeting. But, out of every 10 dose justification reviews that I've done over the recent months, 90% of them still try to justify a higher dose when there's no compelling reason to go higher. So hopefully this has been helpful to the audience, and I look forward to some additional questions with you, David.
Yeah, so we definitely have some time for questions, and I will start by asking, so this is an interesting one. What's the difference between Project Optimus and adaptive design in clinical trial when you can change the dosing strategy according to the current results?
So I talked about this a little bit earlier. So a lot of adaptive designs are still very heavily weighted on safety. But I do think that there are some adaptive designs that incorporate efficacy into it, or maybe a pharmacodynamic marker. But again, if that marker is not correlated to efficacy, that's a little harder to justify the adaptive designs. But I do think that adaptive designs have a tendency to shunt people into certain doses based on whatever you've defined them as long as those definitions include, for your Bayesian approaches, include efficacy, then you've probably addressed Project Optimus a little bit throughout that. But the other thing I want to mention is that a lot of these Bayesian designs have a tendency to enroll a lot more than 3 people per dose level. And so that is also, I think, a little bit of what Project Optimus is trying to do, is getting away from making decisions on N of 3. Yeah.
Great. Another question, should Optimus be applied to small molecule as well?
Of course, I think it probably was birthed by small molecules, and the drug that I was speaking of at the very beginning in 2013 that kind of made me have an aha moment. What are we doing? Why are we doing it this way? Was a small molecule tyrosine kinase inhibitor. Yeah.
Okay. And another question, what does Optimus mean for combination therapy? So will there be better recognition of potential PK or PD interactions?
Yeah, combination therapies definitely make this trickier. And I think it's even harder when you're working with a monotherapy agent that does not confer efficacy, and then how you take that into the combination setting. It gets even murkier when you're using a new combination agent. So you're using your own PD-1 instead of using an approved PD-1, because then you actually have to end up optimizing 2 things instead of just one. The simplest setting, I'm not going to talk about all the complicated ones because there's not enough time. In the simplest setting, you would optimize the monotherapy first. And then ideally, when you put that into combination, the combination agent would have already been optimized. And so, you would obviously start with a safety run-in, but you should be relatively off to the races with regards to that. But you can use PK, PD, but a lot of it's going to go back to whether or not your monotherapy has been optimized for efficacy.
Okay, and another question, how applicable is Optimus to biologics as to small molecules? And how applicable is Optimus to biologics, including gene and cell therapies, as to small molecules?
Okay, so CEDAR reviews both the biologics as well as the small molecule. So Project Optimus is an oncology center of excellence. And I think that while OCE does have some purview with regards to the cell therapies and the gene therapies and the oncolytic viruses, those are technically handled more by CBER. If you were to ask me what Project Optimus is applying to all oncology drugs, I would say that it's probably more hyper-focused on the small molecules and biologics than it is on the oncolytic viruses and the genes and the cell therapies. That's not to say that those people get a free ticket for dosing and dose optimization. It's just that, you know, the majority of the agents that we're developing these days are small molecules and biologics. And so I think that we got to fix the large problem first before we kind of start going into these other agents, which are still relatively new and novel. And I think we're all still figuring them out. TKIs and biologics have been around for years. It's time that we start thinking a little more logically about how we develop these drugs.
So how do you best juggle the hunt for proof of concept with the principles of Project Optimus?
Well, I think that Project Optimus and proof of concept are actually together. You know, Project Optimus makes your proof of concept hunt more reliable and more believable. So being able to show that your drug has efficacy at one dose level, but does not have efficacy as dose levels are lower, or vice versa, shows you without a randomized comparator that your drug is conferring benefit and what you're seeing is real. So I think that, you know, this is like proof of concept on steroids. So, you know, some of the stuff that we see in these early 3+3 designs where we see response rates, you know, sometimes it's very difficult to tell whether or not they're spurious or they're believable. And a lot of it comes down to the fact that those studies have a tendency to also incorporate a lot of different cancer subtypes, so it's hard to see trends. So I think that these 3+3 dose escalation studies may help us understand proof of hope that the target is working and that we're getting some efficacy, but that Project Optimus and dose ranging is what's really going to solidify proof of concept.
Interesting question coming up. So for drugs already in phase 3, are we expected to go back to phase 2, or is it fair to expect a post-marketing commitment to adjust dose?
That's the one I was hoping wasn't going to get
the
answered.
$1 million question.
David, the $1 million question. So for drugs that are already in registrational efficacy studies, I think it's really important that you go back to your current data to review what you have to see whether or not your dose justification makes any sense for Project Optimus. And if not, I think it would be really important for you to go have a conversation with the FDA to discuss how either additional information can be gathered that's outside of the phase 3 study to avoid a potential refuse to file or a complete response action. I do not know. I mean, I think the agency is in a really tough position right now, because if they continue to approve drugs based on single arm trials at one dose level with post-marketing requirements to optimize dose, then we really aren't changing what's happening in the landscape of oncology. It's like they have to be consistent. And so I think that this will be a very interesting year for everybody. I don't know whether or not people that are in phase 3 are still going to be able to file, or if they're going to have to go back and get more data. I've seen a little bit of both.
Time will tell, as they say.
Yeah.
So another question from the audience: what are your thoughts on the impact of this requirement on orphan oncology indications without adequate standard of care?
Oh, that's an interesting one. I mean, I feel that most of oncology is relatively orphan anyway. Everyone's starting to go down these little wormholes of different genetic mutations and resistance mechanisms. And so everyone's pretty much orphan. The interesting thing that I find with orphan drug development, both in oncology and outside oncology, is that everyone thinks that because the patient population is so limited, they don't have to do good drug development. And that's not really the case from the agency side. Like, you still need to make sure that the patients that you're enrolling and the dose levels that you're studying are going to provide you, in even your small population, enough data to make decisions. And so I think that if you have 50 subjects in your orphan population to play with during development, you need to think about how you're going to utilize those 50 subjects. Are you going to stick them all on one dose? Hopefully not. Ideally, you would split them between 2 dose levels. You could very easily take I think there was a drug approved last year. It was very interesting in graft versus host disease that took 2 different dose regimens in a single arm trial with no comparator and got approved. And it was relatively, I found interesting, and no one's talking about it, but I think that's where orphan drugs are probably going to end up going. The ones that don't have a good comparator is where the agency is going to prefer you to study.
Okay. Next question. How will the FDA caution sponsors in terms of dose selection? And will this come at a pre-IND meetings, at the IND filing, or will they mandate an EOP1 meeting?
So I think the comments are coming throughout the spectrum of the development. I mean, I think if you're not having end of phase 1 meetings before you're moving on to either dose finding, or a lot of times in oncology, your end of phase 1 meeting is the last time that you touch the agency until you come in for a pre-NDA or pre-BLA. So I really hope you're having that meeting. But the touch points would definitely be into phase 1 or into phase 2 to have those conversations. And if you don't have those conversations, what I'm seeing is protocols being put on hold until compelling dose justifications are submitted to the agency for review. So that's definitely a possibility if people feel that they either don't want to have that stopgap and go into the FDA to talk about it. They're just going to, fly through. Hopefully no one pays attention. That is a risk that you put into your program if that's the approach that you want to take.
Gotcha. Next question. Can dose optimization in one indication be used to support dose selection in another indication? And what data, clinical, non-clinical, could support this approach? Yeah. And would this approach cause challenges at filing?
Yeah, no, and I think that's a lot of questions, and those are a lot of really good questions. And I think that this is going to really be driven by your non-clinical and how your dose justification panned out in your first indication, or your first therapy that you're taking forward into the clinic. So I would hope that we don't need to do this for every single indication, because that's going to be incredibly burdensome. But I would think that once you've established that responses are ideal at one exposure level or one dose level versus another, that would be kind of where you would start for the rest of your therapies. And again, like I said, the non-clinical studies will be really important to help provide some understanding, because if you have the same tumor growth inhibition across 3 doses and 3 different tumor models, then ideally that would translate into the human state as well.
Okay. And Julie, we'll take one more question. We want to be respectful of everyone's time and wrap this up at noon. So last question, how does this apply to master protocols where a wide number of drug combinations and or disease types are evaluated?
Yeah, so those master protocols should ideally be only enrolling dose levels that have been optimized, is my guess. Master protocols are probably not the best place to optimize a dose. I guess unless you include multiple dose levels within your master protocol, you might be able to get some information out of those types of approaches, but we'll have to rethink how we view those bucketed and master protocol approaches. Yep.
Well, thank you, Julie, for this lively and informative discussion. You're awesome. We appreciate your time and expertise. And on behalf of NASDAQ and Certara, I want to thank everyone who joined today's event, and certainly have a great rest of your day. Thank you.
Thank you, David. Bye, everyone.
Bye.